Yes.
So hi everyone, my name is Kingsley Garrett. I'm a rising senior at Hampton University. For the past few weeks, I've been a summer intern for the NABA Lab at the University of Illinois Chicago through the NIH's summer internship program. And so my project is Matrifold, and it's building a resource of structural data for extracellular matrix proteins from the protein data bank. And so to begin, for those who aren't familiar with the extracellular matrix, it is a meshwork of proteins and glycosaminoglycans deposited by the cells. And so it plays an active role in influencing cell behavior, such as shape, function, proliferation. And it's a complex composition, and our understanding of its organization is continuously evolving. And so within the ECM, there's various proteins that are large and soluble and often cross-linked, which contribute to their mechanical and biochemical properties, such as providing elasticity and rigidity to tissues and relaying signals by binding to cell receptors or modulating growth factor signals. And so highlighting this image on the right, this is a tissue sample from small intestine taken from HuBMAP data set. And so we can see the assembly of the ECM within the tissues. And as the deposition of the individual ECM components are deposited by the cells, it forms these complex networks. And these networks are built upon protein-protein interactions, which is why these interactions are crucial for the assembly and the function. And so to delve deeper inside the specific genes and proteins, there is about 1,000 genes encoding for ECM proteins, which is collectively known as the matrisome. And so within the matrisome, it's divided into two divisions: the core matrisome, which is collagens, proteoglycans, and ECM glycoproteins. And matrisome associated includes the ECM affiliated proteins, secreted factors, and ECM regulators. And so each group plays a distinct role, but also complementary roles. And the structure and the function of the ECM contributing to its complexity and also its versatility. And so what does the NABA Lab actually focus on? It focuses on investigating, of course, the extracellular matrix and also its interactions through a combination of experimental methods, proteomics tools such as mass spectrometry and computational analyses. And so their main goal is to have a better understanding of the extracellular matrix and what roles it plays in causing various pathologies in order to help with developing new therapeutic approaches. And so within the HuBMAP consortium, it's part of the demonstration project titled Leveraging HuBMAP Data to Build the Human ECM Atlas. And so they are especially looking at studies that involve interaction networks, especially pertaining around to the matrisome and how these networks contribute to the mechanisms within the ECM, such as remodeling and the spatial arrangement. And so moving on to being able to understand ECM biology, it requires having detailed structural data. So a dogma that we're all familiar with is that proteins are made of amino acids, and these amino acid sequences are determined by the genes that code for them. And so we want to study these structures to observe conformational changes because the changes that these proteins undergo can affect their function. So an example of this information that we're looking at that can help us with protein interactions could be a collagen. And because its unique triple helix structure, it consists of 3 polypeptides. And so within these polypeptides, they're wound together, and each chain is specifically coded for by a gene. And this also, in turn, helps with the proper assembly and the stability of the helix, which determines its structure, and the structure determines the function of which it has an extracellular matrix. And so referencing this image once again, we can see that, again, this is taken from the HuBMAP data set, that the collagen is fully assembled into the ECM, and that it has a network that is being supported and surrounded by the cells. And so we want to be able to define and understand the steps that come between having a single unit structure such as this and to becoming a full assembly. And so that is why we're looking at the structure, because it determines the function, the interactions, and the roles in which it plays in extracellular matrix. And so that brings us to our main objective of our curation efforts, which is to build a resource by mainly curating structural data of the extracellular matrix proteins from the protein data bank. And so what is the protein data bank? It is a database that provides three-dimensional structures of biological molecules. And so the two main reasons why we're using the protein data bank is because first, it's an open access resource that we can use. And secondly, because it provides experimentally validated structural data of the extracellular matrix proteins, especially ones part of the core matrixome, matrixome-associated factors. And so with this, PDB offers comprehensive information on the entire biological assembly, so how multiple protein molecules interact and function together. And this can help us with our understanding of the structure and the function of the ECM proteins, which is essential for our research of the extracellular matrix. And so moving on to our methodologies that we used, first, we retrieved the raw data of the matrixome proteins from the PDB. And then secondly, we populated the metadata that came with PDB into columns and organized it and managed our data sets within Excel. And then finally, our main curation efforts was centered around determining the structural type and how to classify each protein into those structural types. And so first, to begin delving deeper into the first process, which was retrieving the matrixome proteins, we had a total of 518 entries total. And so with this, the distribution, the categories, the entries were distributed into matrixome categories. And so we saw that majority of the proteins were actually lied in the ECM affiliated proteins, ECM regulators, and the secreted factors, which was fallen into the matrixome division, as I mentioned earlier, in comparison to a small number were part of the core matrixome. And this could be mainly due to the fact that core matrixome proteins are insoluble in nature in comparison to matrixome-associated proteins are more readily crystallized. And so with that also being said, we saw that within the 1,027 genes total of the human matrixome, we only had 91 corresponding from our 518 entries. And so this was a very low percentage in representation coming from our data set. And so this could indicate that not all ECM-related genes are represented in our data set, and we have the potential to expand it. And then moving on to the second part of our process, populating the metadata columns. And so we organized our metadata in Excel, and this is what the structure would look like. And so first, starting off with the PDB ID, each ID references a unique protein structure. And so this will be the reference ID for each one within our entry list. And so this is where it'll be located on the website, the database. And then next, an important part would be the structural title. So this gives a slight description of what we're going to be looking at. And then next, the way that the structures were determined, the methodology used, and for this one was X-ray diffraction. And this was like one of the most common methodologies used to determine the protein structure throughout the PDB database. And then finally, another important one was the expression host, which was located here. And so finally, the structure type and how was that determined and what are we looking at for that? And so that includes our main efforts of our curation. And this is what we're focusing on, how we chose different categories and different criteria set in place to determine the structural type. And so looking at that, we had 4 criteria set in place: title, size, binding, and number of entity IDs. And then we also had the 3 main protein type structures. So it was multimer, fragment, and complex. And so to begin with, looking at the title, the structural title, multimers were absent of key terms. And so this means that there was no words that were outstanding to us when looking at the title, like these were not as significant compared to fragment and complex. But when looking at fragment, the structural title usually gave us a hint of what would be a fragment because it included domain or fragment in it. And then for complex, it included complex or complex width. And then moving on to size, multimers are going to be whole protein structures. So this means that they were full-length sequences or structures. And so from the PDB, we compared the sequences given there to the one in UniProt to make sure it was equivalent or equal to. And then for fragment, it was only a portion. So an example of this would be a collagen will only have 1 alpha chain instead of having 3. And then for complex, it can be both. So this means that the complex could be a fragment and a multimer together. I mean, a fragment and then a full-length protein together, or it could be just 2 fragments in complex with each other. And so this brings us to the binding part as well, because the difference with this is multimers usually had nothing bound to it, meaning it was because it stood alone as a full string structure, while fragments usually are only bound with an ion such as calcium or zinc. And then complexes, they stood out because it was a protein and another protein together and binding as well together, or it was a protein and an polysaccharide bound together. And so finally, our last criteria that we were looking at was the number of entity IDs. And so since multiples and fragments stood alone, they only had one entity ID describing the molecule that was being shown. But for a complex, of course, like I said, since it was multiple molecules bound together, of course, it's going to have multiple entity IDs to describe each molecule, describing its sequence length and things of that nature. And so moving on to the data that we had for that. So within the structural classification of the matrixome PDB entries, we saw that our Lowe's representation was multimer. So only 71 out of the 518. And this low representation could be due to the fact that it's hard to get solubilized ECM proteins, especially full structured ones. So this is a reason why this is such a low number. And then for fragment, it was about 36% of our data, and then complexes being 50% of our data, as well as when we distributed it into the matrixome categories, we saw that fragment actually had a larger portion contained in the core matrixome, while complex and multiples were still seen as being more abundant in the matrixome associated. And so finally, when we look at the most important or valuable insights, we can go to complexes, because we're looking at these protein-protein interactions, because when ECM proteins undergo conformational changes or interact dynamically with other molecules, they are having complex interactions. And these interactions are key drivers of ECM dynamics, such as remodeling or changes in the ECM composition organization. And so these complexes were going to give us the most valuable insights into our data, looking at these protein-protein and protein-ligand interactions. And so with that being said, moving forward, first part of the NABA lab, we plan on continuing our efforts and having meticulous curation of structural data. So this means that we can cover a broader range of ECM proteins, because as I mentioned, we had a low representation of our genes. It was only 91 genes in our data set out of 1,027 genes total. And so with this, we can hopefully broaden our data sets and provide a more comprehensive view of the ECM landscape, as well as complexes, giving us biological relevance and how these interactions can help us understand the ECM dynamics, as well as our focus in the matrixome projects, as it aligns with NABA lab's goals of comprehending ECM mechanisms, as it plays a role in both health and disease. And so not only will this protein level evidence help with the NABA lab, but also it will help with HuBMAP consortium. So as we know, HuBMAP, their primary goal is to have an open access global platform to map trillions of healthy cells in the human body. So given that the ECM is often understudied, the structural data that we derive from the matrixome proteins from the PDB database can hopefully enhance our understanding of ECM biology, but also this protein level dynamics can contribute to HuBMAP data sets, as well as incorporate them into helping create comprehensive tissue maps. And so with that being said, I've had a very enriching summer, and I've learned a lot. To begin with, the interrogation of databases. So of course, HuBMAP, I was able to look into the data portals and the data sets and retrieve information about ECM related data sets that pertain to my project. And then PDB and UniProt, I use these two to provide me with structural data for my curation. And then of course, the actual creation process, I was able to use Excel to organize, manipulate, and manage my data sets. And I learned the process of curating data, and then also able to create data summary graphs to visualize to my audience the key findings. And then of course, quality assurance. So it was very important to have thoroughness and precision every step of my research process, as well as checking for the integrity of my data and making sure it was accurate. And then finally, being able to work with such a diverse team and contribute to my learning process. And of course, communication is always essential to have successful collaboration research. And so with that being said, all in all, I would like to express my sincere gratitude to the NABA Lab members for their invaluable support and their guidance throughout this summer, as well as the opportunity to contribute to such an important project. And then, of course, I'm deeply grateful to the HuBMAP consortium for providing me with this opportunity this summer. And then finally, last but not least, I would like to thank my home by the sea, Hampton University, for preparing me for a role such as this. And thank you all for your attention. I'm open to answering any questions.